I'm going to introduce Elena Pozzi. Unfortunately, Elena couldn't make it at short notice, so she has kindly recorded her presentation. And then after that, we'll be having some top tips from Emmy and Ethan, so some more practical guides onto how to get everything set up. So,
Hi everyone, my name is Elena Pozzi and I'm from Origin and the University of Melbourne. Today we'll be talking about neurofeedback training in young people with depression. I'm very happy to be speaking to that in this session, even though I could not be there in person. First of all, I don't have any disclosures to make. I would like to start by mentioning why we're doing this work. Depression is incredibly... Hi everyone, my name is Elena Pozzi and I'm from Origin and the University of Melbourne. Today we'll be talking about neurofeedback training in young people with depression. I'm very happy to be speaking to that in this session, even though I could not be there in person. First of all, I don't have any disclosures to make. I would like to start by mentioning why we're doing this work. Depression is incredibly common. Chances are that each of us has either experienced it personally or knows someone who has. In Australia, 1 in 7 people will experience depression at some point in their lives, and in young people this number goes up to 1 in 5. Together with anxiety disorder, depression is the leading cause of disability in young people and the leading cause of suicide, which is the #1 cause of death among young Australians. Despite this immense burden, up to 60% young people don't respond to current treatments. And this makes one thing clear: we urgently need new non-invasive approaches to support young people with depression. Depression is marked by difficulties in emotional regulation, including a persistently low mood and a reduced ability to experience pleasure. These emotional disturbances are likely rooted in altered brain function, with disrupted amygdala response to emotional stimuli being one of the most consistently reported findings in depression research. A 2019 review found that most neurofeedback studies aiming to improve emotional regulation have focused on mental imagery involving autobiographical memory, so memory of personal events, and I return to this shortly. The amygdala has been one of the most frequently targeted brain regions in this context, with the majority of studies aiming to upregulate amygdala activity in response to positive emotional content, and specifically positive memories. Much of the existing FMRI neurofeedback research in depression has focused on upregulation of amygdala to positive memories. The study published by Young et al. In 2017 is the first randomized controlled trial that included a control group where subjects received feedback from a region supposedly non-implicated in emotional regulation, the interparietal region. The study found the participant in the experimental group successfully learned to upregulate amygdala activity during the recall of positive autobiographical memories, and changes in amygdala activity during the transfer run, which is when no feedback was provided, were associated with reductions in depressive symptoms. Since then, several studies have been published in depression. However, only few of those included a control group, and very few have explored this approach in young people. Most of the studies use autobiographical memories as stimuli, and why is that? We believe that memory is important in depression because it plays a central role in how individuals process and regulate emotions. In depression, these memories are often overgeneral, negatively biased, or difficult to access from a first-person perspective, and this can reinforce low mood and hopelessness. By helping individuals recall specific and positive autobiographical memories, we might be able to support more adaptive emotional regulation, and this is where neurofeedback offers a unique opportunity. BOOST is a study conducted at Origin and the University of Melbourne and stands for Evaluation of Brain Training to Boost Mood in Young People with Depression. The primary aim of the study is to assess the feasibility, acceptability, and safety of the neurofeedback protocol for young people with depression using an ultra-high-field MRI scanner. The secondary aims are to explore whether young people can learn to increase amygdala activity in response to positive memory, and whether these changes are associated with reductions in depressive symptoms. With this study, we hope to collect preliminary findings to later support a larger randomized controlled trial. We will recruit 20 young people, aged between 16 and 25, experiencing current moderate to severe depression and not experiencing psychosis or mania. Participants will be recruited through Origin Headspace centers and will undergo first through a screening to make sure that they meet eligibility criteria. If eligible, the participant will be enrolled. The baseline visit will include the first MRI and a neurofeedback session and a battery of self-report questionnaires. During the neurofeedback task, participants will be instructed to recall specific positive autobiographical memories while attempting to increase amygdala activity. Participants will then return 1 week later for a second MRI and neurofeedback session. And follow-up questionnaire will be completed online at 2 time points after the second MRI, 1 week and 1 month after the second MRI. And at the end of the study, participants will also complete a debriefing questionnaire to address their satisfaction and engagement with the study. Questionnaires will measure depressive symptoms, anxiety symptoms, difficulties in emotional regulation, and autobiographical memory. One of the first questions that came up in designing the study was around mental strategies to upregulate amygdala activity during memory recall. Previous studies have often reported limited details about the instructions and mental strategies provided to participants during neurofeedback tasks. In many cases, studies did not report what strategies participants actually used and found helpful during the scan. Here you can see some examples of information reported in 3 key papers in the field. The information has been summarized, but you can see that it's quite general with regard to the instructions and mental strategies provided and used by participants. These are some more specific questions we had, including how many memories should a participant prepare in advance, how much feedback should we give them, and should we suggest specific strategies. Basically, we were asking, how do we help participants to achieve the task? So what people will actually be asked to do? In Boost, we will use prompts from the autobiographical memory task to help participants, first of all, come up with at least 3 memories before the task. And then feedback will be given in terms of how specific the memory is. So participants will be given a minute to retrieve a specific memory in response to a cue word. So example cues would be positive words. So for example, interesting, independent, proud, and friendly. And then we will look for specificity. So for example, day, time of the day, when the event has occurred. So what strategies could we suggest participants to help them upregulate their brain activity while they recall positive autobiographical memories? I'm very keen to hear your thoughts. So I'm going to give you 1 minute to scan the QR code and make your suggestion in the form. Hopefully at the end of this presentation, my lovely colleagues will be able to bring up the responses. Thank you. So what I thought is that some example strategies could target what we know to be altered in autobiographical memory recall in individuals with depression. So for example, we might suggest individuals to use vivid mental imagery. So recall a specific detail and emotionally positive memory, such as a birthday, holiday, or a personal achievement. We could encourage them to engage all senses. What did you see, hear, smell, or feel? Another thing is that we could suggest to use perspective taking, so recall the event from a first-person perspective. We could also suggest to use self-affirmation and positive self-talk. So use internal dialogue to reinforce the positive meaning of the memory. For example, phrases like such as, I felt so proud in that moment, they show me how capable I am. We could also suggest them to focus on the relational aspect of the memory, so emphasize the social connection and closeness in the memory. So for example, being with friends or family. And why it's important to reflect on strategies. But basically, we really want to know what we are targeting. According to the consensus on the reporting and experimental design of clinical and cognitive behavioral neurofeedback studies checklist, that was a very long name, mechanism influencing neurofeedback outcomes can be grouped into 5 categories. Neurofeedback specific, so effects that come from training the targeted brain region. Neurofeedback non-specific, so effects tied to the neurofeedback context but not specific to the targeted brain region. General non-specific, broader effects from cognitive training or placebo. Repetition related, So improvements that come just from repeated testing, and natural changes due to spontaneous recovery or development. And moving forward, we want to design studies that can clearly disentangle these overlapping influences and help us understand which mechanisms are driving the outcomes. So this checklist is designed to promote reproducible research and transparent reporting in neurofeedback studies. So one of the items included is psychosocial factors, including the self-efficacy and strategies. So strategies, reporting on whether participants were provided with a strategy, and if yes, what strategies is considered essential. Another question that came up was around the control group being a small feasibility study. Trambayoli published a paper discussing the different types of control group for neurofeedback, which are essential if we want to demonstrate that neurofeedback can outperform mental rehearsal and placebo factors, so the other mechanism that we have seen in the previous slide. As you can see, we can go from no control, which are the columns on the left, to within subject control, for example, using bidirectional regulation control. So in this case, participants perform both up-regulation and down-regulation of the same region, so the same participants. However, this might not always be possible. For example, in our study, we probably would not want to ask young people with depression to remember negative memories and try to down-regulate amygdala activity in the scanner. So this might bring up some ethical concerns or might not always be possible. The placebo control group, so the orange columns, include, for example, providing participants with an alternative feedback, so signal from an alternative brain region that we think is not involved in the key mechanism. And lastly, the mental rehearsal or strategy-only control group can be used to determine whether applying a self-regulation strategy alone without the feedback can induce a similar changes as the neurofeedback. So in this condition, participants are instructed to use a specific, clearly defined strategy, the same as the neurofeedback group, but they receive no feedback. This setup can be much more economical and easier to implement because it can be done outside of the scanner. Even though mental rehearsal alone cannot fully rule out placebo or motivational effects because it lacks the feedback component, it can at least inform whether the regulation strategy, the behavioral strategy alone, can account for the changes. And this is what we plan in the end to do in Boost, given the, as I said before, limited funding. So we will have a group just practicing retrieving positive and specific memories to see whether this can account by itself for changes. And if that's the case, that would make the decisions around the whole study different, because obviously we need to justify the cost of the MRI. And this highlights again the importance of clearly defined strategies so that different groups can use the same strategies. For the next few slides, I would like to acknowledge Ethan and Amir because they helped prepare this content. And finally, in designing Boost, we carefully consider which rounds to include in the study. So we base our protocol largely on the original design by Young et al., which we are closely replicating. However, their full task lasted approximately 52 minutes, not including the structure scan, the check-ins with the participant, and the data analysis time. So given that we needed to ensure feasibility and tolerability for younger participants, we made the decision to shorten the task duration, but we also wanted to retain core elements of the original design. So we initially considered several design options, including removing the first transfer run. So at the top is the original version. The second one is one of the options we consider. We consider removing the transfer run because the main aim of the study was to assess feasibility. And at first of all, we wanted to demonstrate the participant can undergo the protocol and show changes in the neurofeedback run compared to baseline. These were our other options that we consider, such as removing the baseline. This is when participants do the same task but don't receive any feedback. And this was just to show that previous paper showed strong signal changes already after the second run, which provided a rationale for not including a third run. Ultimately, we decided to include a baseline run 2 neurofeedback runs and a transfer run. And the transfer run is particularly important because it tests the central hypothesis of neurofeedback. The participant can learn to voluntarily regulate brain activity through the feedback-based training. And to support this, we need to demonstrate the participant can continue to modulate the brain signal even after the feedback is removed. And this really would indicate that the effects reflect genuine learning and not just transient changes that are driven by the feedback. So to conclude, I share some of the key questions we consider while designing this ongoing study. As the project progresses, I hope to provide more definitive answers in the future. One key takeaway so far is the importance of reporting as much details as possible, not only on the neuroimaging side, but also in relation to behavioral changes. And this is really to support reproducibility practices in this relatively new field. Here are just some resources you might find useful. So the checklist I mentioned and the article on the different type of control group that you can include in your study. And finally, I would like to thank all my collaborators that have helped me in this project so far, from the University of Melbourne, Origin, and ACU. And a special shout out to Amir and Ethan, who have been really instrumental in getting this study off the ground. And thank you all for listening.